The purpose of this lab was to observe the reaction between two aqueous solutions, copper sulfate and potassium hydroxide, in different mole ratios. Our objective was to determine the limiting reactant at different ratios by measuring the height of the precipitate formed.
Stoichiometry is the study of quantitative relationships between amounts of reactants used in products formed by a chemical reaction. By using the equation given, 2KOH plus CuSO4 produces CuOH2 plus K2SO4, we can see how concentration takes place in several of the tubes as the ratio of solute to solvent is unequal. Concentration is measured in either moles or volume. The mole ratio illustrates the relationship that exists between amounts of reactants and products. Reactants do not always form the amount of products as specified in a simplified balanced equation. Typically, there is excess of one or more reactants, therefore the reaction continues until all or one reactants are used, causing there to be a limiting reactant. A limiting reagent, also known as a limiting reactant, is what is fully consumed when the chemical reaction is complete. Without the reagent, the reaction cannot continue, therefore limiting the amount of product formed. A precipitate is a solid that forms from a liquid.
The materials used in this lab were the aqueous solutions of potassium hydroxide and copper sulfate. There are also 8 test tubes, a burette, spatula, metric ruler, and water.
KOH, also known as potassium hydroxide, was mixed with CuSO4, also known as copper sulfate, in 8 separate test tubes. We then wrote a balanced equation for the double displacement reaction. The equation we created was 2KOH plus CuSO4 produces CuOH2 plus K2SO4. To proceed with the reaction, we dispensed 5 milliliters, also known as 0.005 moles, of CuSO4 into each test tube. Then we added a certain amount of moles of KOH to each test tube according to the volume chart we were given. Water was then added and mixed with a spatula so that each test tube would have a solution of 13 milliliters. We stirred each test tube with the spatula and let it sit for 15 minutes while we timed. Finally, we used a metric ruler to measure the height of the precipitate in millimeters in each test tube.
While performing the lab, we saw a vibrant blue crystal-like structure at the bottom of the first 4 test tubes. The 5th test tube appeared to be a well-mixed light blue solution. Test tubes 6 through 8 were also blue solutions, but they had excess clear liquid sitting at the top.
During the lab, the mole ratios were uneven with the exception of the 5th test tube. The mole ratios were 2 to 1 because of 0.2 molar of potassium hydroxide and 0.1 molar of copper sulfate that was added to the burette. In all 8 test tubes, 0.0005 moles of copper sulfate were added, being converted from milliliters. However, there was different amounts of potassium hydroxide in each test tube. Tube 1 held 0.0002 moles, Tube 2 held 0.0004 moles, Tube 3 held 0.0006 moles, Tube 4 held 0.0008 moles, Tube 5 held 0.001 moles, Tube 6 held 0.0012 moles, Tube 7 held 0.0014 moles, and Tube 8 held 0.0016 moles. The lab contained 8 test tubes with various amounts of reactants. All of the test tubes had limiting reactants except for the 5th test tube. The 5th test tube had an equal mole ratio of 2 to 1, containing 0.001 moles of KOH and 0.0005 moles of CuSO4. Therefore, both reactants were used, leaving no excess and having no limiting reactant. For the first 4 test tubes, the limiting reactant was KOH and the excess was CuSO4. For example, in test tube 1 there was 0.0002 moles of KOH and 0.0005 moles of CuSO4. Tubes 6 through 8 had a limiting reactant of CuSO4 and KOH was the excess. For instance, in tube 6 there was 0.0012 moles of KOH and 0.0005 moles of CuSO4, showing the unbalanced mole ratio.
One of the errors that may have occurred during this lab was that the tubes were not all filled to exactly 13 milliliters. This would affect our results because the heights of the precipitate might have been different due to the amount of water added. Another error could have been that we did not let the test tube sit for 15 minutes. Instead, we left it for 10. This is an error due to the fact that we did not let the solution fully react. therefore giving us wrong measurements of the precipitate. A suggestion to better our lab results could be to set a timer to correctly time the amount needed for the reaction to occur.